The bodily reaction I decided to focus on is the binding of diatomic oxygen to the heme group of hemoglobin, the protein found on red blood cells that transports oxygen around the body. Without this process, the blood's ability to carry oxygen would be severely reduced, and humans would not be able to survive. This reaction is relevant to my field of marine science because almost every fish on Earth uses hemoglobin. The reaction centers around the iron atom in the center of a porphyrin ring, the key functional structure of this process. 4 of the 6 bonding sites on the iron atom are taken up by the nitrogen atoms on the porphyrin ring. Due to a raising of pH and/or other factors, this group, as it appears within the hemoglobin protein, can become more likely to accept a diatomic oxygen molecule in exchange for releasing a CO2 molecule. When it does this, the structure of the ring shifts from a bent form to a planar form, which pushes the histidine group, which connects the heme group to the rest of the protein, farther away. This changes the shape of the whole molecule in a process called cooperative binding, and this makes the other heme groups more likely to accept oxygen. When the complex encounters an area of the body with lower pH, this coerces the process to reverse and release the oxygen molecules, exchanging them for CO2.
For this section of the video project, we're going to be talking about hydrolysis of DNA. So DNA is made up of monomer and chains of nucleotides, and nucleotides could be guanine, cytosine, thymine, or adenine. And these are joined by phosphate diester links, and that's where the O chemistry of it comes in. So these nitrogenous bases link in their chains, and to make that DNA helix strand, they're formed by hydrogen bonds as well. So the important process to break the chain is to either change or repair this DNA sequence, and that's really important for evolutionary purposes, just to be able to fix that or change it when needed. And so hydrolysis is literally, lysis is like the breakage through nucleases, and the breakage of those bonds where water attacks and eliminates the phosphate, and then that OH group will reattach where that elimination takes place, and the DNA reconfigures.
This is stomach digestion and polypeptide breaking reactions, and I'm Allison Fayard. There are 3 main types of digestion that occur in the human body: protein digestion, carbohydrate digestion, and digestion of fat. Digestion of proteins occurs in the stomach and duodenum and utilizes 3 primary enzymes: pepsin, trypsin, and chymotrypsin. Carbohydrate digestion utilizes salivary and pancreatic amylase to break glucose molecule bonds. And finally, fat is digested by lipase from the pancreas, which breaks down short-chain lipids into diglycerides. Protein digestion and absorption require complex chemical reactions. Protein digestion begins in the stomach because of the low pH of stomach acid, which denatures it. This means the protein will begin to unravel. In the image to the right, you can see the blue coiled protein unwinds into a strand of amino acids. When the long strands of amino acids are broken into smaller segments, they are called polypeptide chains. In the intestines, dipeptides and tripeptides are formed as the enzymes from the pancreas and intestinal lining break down the polypeptide bonds. Finally, dipeptides and tripeptides are separated and result in individual amino acids that are now available for absorption.
So for my reaction that takes place in the human body, I will be talking about the metabolism of alcohol, which is important to humans because although not everyone consumes alcoholic beverages, we all eat foods that contain small amounts of alcohol. This is important to my field, major of marine science, because this is a good example of processes that are carried out in the liver, much like those that would occur in the livers of marine mammals or fish. Now, for this process, ethanol is either reacted with alcohol dehydrogenase or with the CYP2E1 protein, both of which need to be activated by turning NADPH or NAD plus into NADH or NADP or NADP into HO, as you can see in the diagram. Now, both of these reactions would form acetaldehyde, which would then need to react with aldehyde dehydrogenase, which also needs to be activated by combining NAD plus and creating NADH. And this process would form acetate, which then can be used by the human body.
In this section of the project, I'll be talking about dehydration reactions. A dehydration reaction is a chemical reaction between 2 compounds where one of the products is water. This occurs when a hydrogen from one molecule reacts with the hydroxyl group from another molecule, forming water. This removes the water from the two molecules, hence the name dehydration reaction. This also causes the two molecules to covalently bond together. This is very important in many natural processes, including the formation of sugars and polypeptides from monomers and the synthesis of proteins. In the case of polymers, a glycosidic linkage is formed between the two monomers. As shown in figure 1 above me, each monomer, such as glucose and fructose, consists of a hydroxyl group. A glycosidic linkage consists of two hydroxyl groups where one hydroxyl group gives both the oxygen and the hydrogen, and the other hydroxyl group gives just the hydrogen, leaving the oxygen to be unbalanced. A bond can then be formed between the two monomers at the oxygen. In the case of proteins, a peptide must be formed first. Peptides consist of two or more amino acids, which forms a polypeptide chain. Amino acids consist of an amine group, or NH2, a carboxyl group, OH and a double bonded oxygen, and a side chain. These side chains vary between amino acids. To form a peptide bond is formed with dehydration synthesis. This is very similar to a glycosidic linkage, but instead of reacting two hydroxyl groups, peptide bonds consist of an OH from the carboxyl group and the hydrogen from the amine group. This forms a water molecule, which is removed, forming a peptide bond. After the necessary amino acids are bonded together to form a polypeptide chain, the polypeptide chain is then shaped into the correct configuration to form the protein. That concludes the overview of dehydration reactions and some of their implications.